I'm Kiminobu Sugaya from the College of Medicine. I'm not going to talk about everything of my research because I'm doing so many things. My major is the stem cell research in the neuroscience area, but I just filed a patent on the COVID-19 therapy and also the detection and so on. So I'm doing so many things. But today I want to talk about a little bit how to diagnose the Alzheimer and the other diseases using your saliva. So I don't have to talk about too much about Alzheimer disease. You may know what the tragic disease and so on. And then these are the old markers for the biomarkers for the Alzheimer disease. And then when you check the amyloid beta and then the phosphorylated tau, as you can see here, the control and the clinical AD, you can see the tendency, but it's not the real biomarker. So then what we can do is we can look into the exosome. I'm going to talk about the exosome later, but if we check those A beta or the phosphorylated tau contents in the exosome, you can see so much difference between a control and Alzheimer patient. Yeah. So what is the exosome? Exosome is the small particles released by most of the cells. And then they're using this exosome as the matter of the transmission of the signal or so on. And then the good thing is that this exosome having everything the cells have in the inside, like a messenger RNA or protein or anything. And then this membrane is the cell membrane. That means the lipid bilayer membrane. So it can go through everywhere in the body. So that's why even this exosome released by the brain cells, you can detect in the saliva. So that's the beauty of this one. And then yet the inside of the exosome is quite protected. And then we can measure all here's talking about the synuclein. That could be a marker for the Parkinson's disease. Already I talked about the A beta and then the phosphorylated tau and so on. So then now we are measuring the inside of the exosome, what's going on to see the brain. So the glioma, it's a very bad type of the cancer, and then the patient usually doesn't survive more than 2 years after get the diagnosis. But still the earlier diagnosis is better, and then now we have a cure for this disease that has been developed in my lab. And then we want to see the early diagnosis. And then already we published this paper in PLOS ONE showing such a patient's cells producing a certain type of the exosome, which contain certain type of the DNA, which has the special insertion. So then by looking at those marker, maybe we can say that, oh, you have this type of the cancer in your brain. And then we check another gene, which is a SOX2 DNA, and then we found several mutations for the polymorphisms. I hear there's some of them. Yeah, it's okay. So then those polymorphisms can be also be used to detect this cancer. Right? And then again, the good thing is that these things can be detected in the saliva. Even we don't need the blood sample to work with. And then how about the protein? That was the PCR for the DNA, right? PCR is great. You can amplify such a target gene quite much. Oops, right? Even the 30th cycle, 2 to 30, that's the huge number of the amplification. But how about the protein? Can we use the PCR to detect the protein? So that's the things we are working on now. And then usually the protein can be detected by the ELISA. ELISA, this antibody having a certain enzyme which make a light, and then we can detect as the light. Are you still there? Okay. Yeah. All right. So then we can put the DNA molecule to this antibody, and then instead of the light, we can amplify this DNA molecule to detect the target, the molecule, right? So that way, we can just need a PCR to detect both gene and then the protein, just one PCR. And then the good thing is already we're developing the PCR machine, which with the collaboration, and then those PCR machine without DNA isolation, and then from the saliva, you can provide anything anywhere by anybody, because isolation, such a tedious step, doesn't require. So then but saying that, at this point, we're analyzing the lots of exosome put together, and then get such a insight of the exosome contents and then amplify. But if you can do the single exosome analysis, if you can detect the single, isolate the single exosome and then analyze all the stuff inside of the exosome, that's the we want to do, because each exosome released by the certain cells. By analyzing the protein on the surface of the exosome, we can say, oh, this exosome coming from this type of the cells, for example, like neurons, right? And then the dopaminergic neurons, we can tell that. And then when we look at the inside, maybe those exosome carrying the signal from those cells, if they got damaged, we're looking at the mitochondrial gene and every other genes, and then we have those markers. So then if we can analyze the single exosome, we can do this, the single cell analysis. That's been done, and that's easy now. Used to be hard, but that's easy now. But here we're talking about exosome, which is almost like a COVID-19 size. Can we isolate those? And then the PCR part, amplification, I wouldn't worry about. Single, the exosome should be able to holding the enough signal to detect. So then what I want to do the single exosome multiplexers real-time PCR, and then the multiple real-time PCR part is already we are developing with the one of your faculty. So then that's been done. Only the thing is that we need to isolate a single exosome and do the PCR in the exosome. Here's just an example. I don't want to stick with this. I don't want you to stick with this, but like a digital PCR, okay? That's one produce the droplets, and hopefully we can have a single exosome in individual droplets, and then we can do the PCR in the droplets, and then they detect those signal in the droplets. That way, maybe we can do the single or the exosome analysis. That's my idea, but I'm also open to any of the idea. And then this is frontier. Nobody has gone before, so that's the things I want to try. Right. So then so far this one this project is funded by the Florida Health Department. Thank you so much.